Hi everybody, welcome to our third vodcast in our matter domain. In this vodcast we're going to spend our time looking at the element carbon, because essentially all biology and all life as we know it so far is in fact carbon based. It builds, it is the foundation of all of our molecules that make up what we are. So why carbon? Well, it's abundant and it's versatile. Okay, two huge pieces, I guess, that go in its favor. If you can look as far as elements go, and these are the main elements of life, we find that carbon being the most versatile because it has four electrons in its outer shell, which means it is capable of making up to 4 bonds with 4 different atoms. So it's tetravalent because tetra 4, right? So this allows for a huge amount of variety. So here's just a couple of ways in which we can see carbon bonding. We have methane and ethane, and you've got a double bond right here between these two, and then you've got, just some other structures. So this is just a little something fun to look at. However, there's nothing overtly special about the building blocks. We have a couple of theories that go along this idea of this life force, this mechanism that they're, theory, nothing special about how life's built. We have the materials, we bond them together, we have it. So we've got Stanley Miller's experiment where he tried to recreate early Earth atmosphere. So he simulated this, putting these conditions together where he looked and had this huge electric shock, and voila, he as a result found that he put together these hydrocarbon pieces. So this experiment began, I guess, a real serious look into how life and our molecules really started to be putting together. So, as far as molecules go, there goes the vitalism, okay? Because we can see it's just a combination of these molecules. So one of the most important aspects of organic chemistry is this idea of isomerism, kind of crazy word, but what we're talking about are isomers. And what isomers are molecules that have the same molecular formula, but they have different structural formulas. So they're actually built differently. This would be an example of one. You can see, and if you counted up the atoms, you'd have the same molecular formula, the structure is different. Another great example is glucose and fructose. Both are C6H12O6, but then if you were to actually look at their structures, they're different. So any molecule more complex than propane will have at least one isomer, which is, again, that adding to that versatility of our carbon atoms. Okay, and this is what we call an emergent property. This is one of the vital characteristics of life, and there's some really interesting things that happen with isomerism. So 3 kinds of isomers I want you to be familiar with. There's structural isomers, same formula, different order. Boom, done. Okay, there you go. There's also this idea of what we call cis-trans isomers. Those of you that might be wondering about the idea of trans fats and stuff like that, this is where that term comes from. So you can see that we have the same formula but different positioning around a double bond. Okay, so it actually alters around the double bond, and so that's pointed out here. You can see 2 X's here on the same side of whatever that is. Here and here, they're swapped. Okay, so they're on opposite sides. So with cis, same side, trans, opposite sides. Then we have this really interesting one called enantiomers. And this is the same formula, but it's a mirror image. So around the central carbon. So instead one might look like this and the other one looks like this. So you get this sort of idea of left versus right and this sort of mirroring that occurs. And so you've got, oftentimes you'll see with organic molecules, this idea of L and D enantiomers. So here's one where it talks about sort of the differences. We've got something like ibuprofen as a medication. You can see that there's 2 enantiomers. There's one that's effective as a pain medication, and if you use the other, its mirror image, it becomes ineffective. And we tend to use these forms, and we tend to only use one of the types of enantiomers, and sometimes our bodies will actually convert molecules into enantiomers. And this is what happened in the 1950s, it turns out, when the medication thalidomide was used to give to moms to help to combat morning sickness. So if you click on the link, you're going to see what I mean by that. So kind of take a second and kind of think about potentially why our bodies can only handle and use one form. I think we'll talk about that in our disco. So here's where a little bit of the fun begins. So I'm not going to do a whole lot of talking here, but I want you guys to be introduced to the functional groups. So between your text, which showcases them as well, and some of the supplemental stuff, this is where I'm going to kind of go through these here a little bit quickly, but you can take your time with them and get to be able to recognize and to understand functional groups, because these are key in helping us understand properties of molecules, and generally at the functional group is where reactions occur to link, let's say, monomers into polymers. So here we go. Hydroxyl. Take a second. Look at it. Look at the structure. Look at what it does. Look at the functional properties that it has. So I'm basically going to kind of introduce them. You know, this is our alcohols, and see what it does when a molecule has this OH group. Carbonyl, double bonded carbon to an oxygen. We have two types of carbonyl groups. You've got ketones and aldehydes. So please note, you've got your ketone, has the carbonyl group within the carbon skeleton, so somewhere in the middle, whereas an aldehyde is the double bonded oxygens at the end of it. Carboxyl group, I know we've seen this before. This is that C double bonded to an oxygen with an OH group, so COOH. This creates an acidic property, and we see it in, primarily you'll see it in amino acids as well as some others, so take a look here. Your amino group, hence the name amino acid, that NH2 group, so nitrogen with two hydrogens. Okay, so take a look there at its functional properties. Sulfhydryl, this is a sulfur bonded to a hydrogen. You're going to find these in really only amino acids. This is where we get our disulfide bridges from. Okay, so these are vital in creating disulfide bridges, since this is where we find the majority of the sulfur that exists in our molecules. Methyl group, CH3, okay, and you'll find these a lot. So take a peek, and yeah, those methyl groups are important. When we talk about methylation in terms of gene control, methylation would be the addition of a methyl group. You've got your phosphates. We know about these when it comes time for ATP. So PO4, this gives a negative charge. So you find these a lot either on glycerols, and you find them on ATP and various other things like DNA and so on. So there's the chunk of the functional groups that I want you to know. So a couple of things. A little difference goes a long way here when it comes to functional groups. If you have just slight changes, okay, whereas like all of our steroid hormones, for example, they're based in cholesterol, so they have a cholesterol four-ringed structure, and then they have offshoots that vary, and both estrogen and testosterone derive from that cholesterol, and just slight differences depending on how much you have, you could get, you know, if you have just the type that's testosterone, so just a touch difference in the molecular structure, having an abundance of that sends you down the male route and male properties, having more estrogen, female. So this entire difference in the physical appearance is due to the effects of these two hormones. Very, very similar in structure, just tiny little differences, as you can see. Got a double bond, and you got an OH group in estrogen. That's it. So, with that being said, there we have our carbon. Really take some time, spend some time with the functional groups. This is going to be, you know, it's an important ability to recognize those functional groups as well as what they do to the molecule. And from here on out, we start to put all this stuff together and really start to dig into the macromolecules. So, have a great night, guys. Catch you in class. Bye.